My research over the past 2 months has been centered around LQTS or long QT syndrome. In this disorder, a longer QT interval results from abnormal activity of ion channels in cardiac cells. This genetic disorder is an arrhythmia that is caused by mutations on up to 17 different genes. I leveraged Geisinger's MyCode database, which has genetic information on 175,000 patients. Since minor LQTS constitutes 10% of LQTS cases, 1 out of every 20,000 individuals is likely to have LQTS through these minor genes. This is roughly 10 individuals in our cohort. After filtering through variants in MyCode, it was found that 200 individuals had true loss of function, protein truncating variants on any of the 6 genes we are studying. We know there can only be roughly 10 people that have true LQTS given the rarity of this disease. Surely all 200 of these individuals cannot have long QT syndrome. This tells us that loss of function on a given gene may not actually be causal of LQTS, or perhaps loss of function may have a very low penetrance. This essentially means that not every individual with a given genotype expresses the associated phenotype. Next, I decided to take a closer look at KCNJ2, one of the 6 genes we are studying, to see if patients with truncating variants actually have LQTS. After identifying 4 protein truncating variants, I decided to take a closer look at each individual with these variants. I made an age-matched control group of QTC intervals to better determine if an individual's QTC is elongated. It was found that only 3 individuals had elongated QT intervals. Patient 1, however, only had 1 ECG. A patient with true LQTS is likely to have had multiple ECGs if the cardiologist found irregularities in the first one. It is possible that the single ECG yielded inaccurate results. Patients 5 and 7 both had multiple ECGs and had elongated QTC intervals. Upon looking at their diagnoses, it was found that both individuals were of significant age, suffered from heart failure, and various other cardiac pathologies. All of these are confounding variables, and it is likely that their elongated QTC interval is from these issues rather than loss of function mutations. Overall, this research elucidated that a presence of a mutation does not necessarily mean that LQTS phenotypes will present. Through an in-depth look at KCNJ2, it was found that protein truncating variants did not cause LQTS, and that cases of elongated QTC were likely unrelated to long QT syndrome.